All right. Hello, everyone. My name is Lee Levitt. I work in Dr. Oliveira's lab, where we use Constellation Pharmacology to help us understand pain. So, the title of my talk, Farming for Functional Neuronal Cell Types, really gets at the heart of what Constellation Pharmacology and what our technology can do. So, pain, right? Pain has been studied for basically ever since scientists have been looking into things. This is a figure from Rene Descartes' study on pain from 1696. And his thoughts on this subject have really influenced the way we think about pain. And simply put, pain is good, right? Everybody feels pain, and this protects us. Pain tells you when your foot is in the fire, and it tells you to get your foot out of that fire, or else you're going to damage your foot. But the problem is pathological pain. Pain that has arisen without a cause, or pain that simply won't go away. For example, when I was a child, my father was logging here in Utah, and there's this thing that loggers call a widowmaker. It's a big branch that's at the top of the tree. And my father was out there logging alone. And what happened was when he was cutting down this tree, this widowmaker fell out of the tree, hit him in the back of the head while he was using his chainsaw, and his chainsaw cut into the right side of his leg. Fortunately, he didn't lose his leg, but he did lose quite a bit of blood. But ever since then, my father has been affected by this syndrome known as pain hyperalgesia. And this is hypersensitivity to touch. This is when a normal touch sensation becomes painful. Another example of pathological pain are patients who have breast cancer and who undergo chemotherapy treatment. There's a specific drug called oxaliplatin, and it's a platinum-based chemotherapy. And oftentimes at the end of these treatments, these patients end up with a syndrome called cold allodynia. This means a normal cold sensation is now excruciatingly painful. For example, these patients, when they pick up a cold glass of water, something you and I would simply think of as just cold, this becomes excruciatingly painful. So what we're doing with Constellation Pharmacology is shedding light on these type of pathological syndromes. And one of the first places that we feel this pain is within the dorsal root ganglion. So the dorsal root ganglion, I'm just going to go ahead and grab my laser pointer so I can talk to you guys. So the dorsal root ganglion sits right outside of the spinal cord, and it sits at regular intervals all along the spinal cord. What I'm circling here are considered where all the cell bodies are. And if we look over here to the right, every green dot is a cell. And these cells send projections clear out into their environment where they sense the pain, the heat, the cold. And then they transmit that back to the central nervous system where it's processed and it's made sense of. But our problem is trying to define which of these neurons sense that pain. And this is a little depiction showing the dorsal root ganglion. These circles here are the cell bodies, and they send the projections out into the extremities. Here they're innervating hair follicles. And so when a hair is touched, right, this is called a mechanical sensation. This is now transmitted back to the central nervous system. So how do we try to understand which of these neurons sense that pain? Well, we use this thing called the primary cell culture. And what this primary cell culture does, if I go ahead and zoom in here, is we break up that system. We break it up into individual neurons. So we take this three-dimensional structure up here, our intact DRG, and we dissociate it and put it in a two-dimensional plane so that we can see everything very easily. You can see that a majority of these neurons are now very separate from one another. And so the outline of my talk today is how we go about classifying these DRG neurons. the dorsal root ganglion neurons, right? These are where our pain sensing neurons are going to be. Additionally, I'm going to uncover for you how we identify unknown neurons within our primary cell culture with known modalities. So what is known in the field? Then I'm going to talk about how we really leverage the transcriptome, and I'm going to touch upon how we use our cell type identification to track disease states. So let's go back to our primary cell culture. Remember, this came from that three-dimensional structure in the animal that we gently broke up and put into this two-dimensional plane. And immediately what we can see is that these neurons can be defined by their size. We have everything from small size, medium size, and even large diameter neurons. In addition to this, we can immediately classify neurons based off of what type of pain they sense. And this can be defined by the neurotransmitter that they release. First off, we can use this marker that is known to the literature for the past 20 to 30 years to identify peptidergic nociceptors. So let's break down that term, peptidergic nociceptor. This means we have a pain-sensing neuron, nociceptor. When stimulated, is going to release a peptide, known as calcitonin gene-related peptide, onto the spinal cord. So that means when you feel a very hot sensation, this specific neuron, these green neurons in our cell culture, are now going to transmit that signal back to the CNS and release a peptide onto the spinal cord. We used a genetically labeled mouse to see this label. And the other type of neuron, which we can easily define, is called non-peptidergic nociceptors. And this is labeled with a lectin. And over here in our image, we can see neurons which are red are considered the non-peptidergic nociceptors. This means they're pain-sensing neurons that, when stimulated, do not release a peptide. Now it's our goal to classify every single one of these neurons into a cell type classification. And right now, this is what we consider to be our current understanding of the cell type classification using our system. And immediately, just based off of these structural identifications, the size, and the label, we can identify which of these neurons now sense that pain. And at this point, I'm going to show you how we use Constellation Pharmacology to further break out and identify these 16 different cell types. And so all of these neurons can be defined by the sensory receptors that they possess. For example, TRPV1. The way I want you to think about these sensory receptors is that they're just switches. They turn on the cell to let the cell know that something has happened. So TRPV1 is going to be this ion, what we considered an ion channel, basically just a switch, that when heat is present, it's going to open up and tell the neuron that heat is present. We also have an ion channel called TRPM8. This is a switch which turns on when there's cold present. And this neuron is going to tell you when this neuron is going to possess the TRPM8, and when cold is present, we're going to know that it's cold because of this protein. And then the other ion channel, which is a bit difficult to explain and hard for the field to get a grasp on, is called this TRPA1 ion channel. And essentially what we need to know for this talk is how it senses a noxious sensation, or it's a sensor of intensity. And so now how do we go about understanding which of these neurons here on the right possess these ion channels or switches? Well, we turn to Constellation Pharmacology. So let's break down that name a little bit. What does Constellation Pharmacology mean? Well, Constellation Pharmacology means that we leverage pharmacology to help us understand the constellation of ion channels and receptors on the surface of these neurons. Our first type of pharmacology that we use are plant-derived natural products. These are going to be molecules that plants produce to tell animals not to eat them. So what do we do as humans? We eat them, mainly because they're delicious. But for example, AITC, this is a specific molecule that's found in wasabi, spicy mustard, and horseradish. So we isolate this compound AITC, and what AITC is going to do is it's going to open up this ion channel, TRPA1, regardless of whether there's a noxious sensation or not. So let's get into Constellation Pharmacology. So Constellation Pharmacology, we start out with loading these neurons with this dye called FIRA2. This is going to indicate to us when calcium has entered the cell. And calcium is going to enter the cell through these ion channels. So simply by applying this plant-derived natural product, AITC, basically putting wasabi on our neurons, we're going to understand which of these cells possess this ion channel. So now what we do is we take an image every 2 seconds as we apply these plant-derived natural products. So the first thing I want you to watch is simply the video here in the background. And what we're going to do is we're going to apply this plant-derived natural product, AITC, and you're going to see what happens to these neurons. So here we go. We apply AITC, and we can see that a lot of these neurons go from blue to a green or red. This means that calcium has entered the cell and that these neurons possess TRPA1. Now if you notice here, cell 2, it's still red. And so the way we capture this information is here in a trace. So just a reminder, we're taking an image every 2 seconds, and this helps us to understand which of these neurons possess these different switches. All right, so now let's go back to our cell type classification. We can now understand which of these neurons have that ion channel. And this further allows us to define one of the 16 different cell types. All right, now let's go back to everybody's favorite plant-derived natural product, menthol, right? This is produced by mint plants. It's thought that the mint plant produces this compound menthol to simply make delicious mojitos. Just kidding. It's probably most likely made by these plants to deter insects from eating it. But we're going to use this menthol and we're going to apply it to these neurons. So now what I want you to keep your eye on is not these neurons, but I actually want you to watch the trace, right? So up to this point, cell 2 has given us a nice big response to AITC. This tells us that this neuron has TRPA1. And now what we're going to do is we're going to apply menthol, and cell 1 is going to be the neuron that responds. Up to this point, we know that neuron 1 does not possess TRPA1. But here when we apply menthol, it's going to respond, and so that means it possesses this ion channel, TRPM8. That means this neuron senses cold. So here we go. Let's go ahead and apply it. And here when we apply it, we get a nice rapid response. All right, and what I want to do is I just want to play this one more time for you, and I want you to actually watch the cell respond. All right, so there we go, and we can see, let's do that one more time. Watch that cell 1 in the top right corner, and here when we apply it, we're going to see it turn red very briefly. Great, so this is what allows us to understand which neurons sense cold. So now we go back to our cell type classification and we can identify which of these neurons sense cold. And I think this is where it gets really interesting, right? Because, let me grab my laser pointer, because now we have a cold sensing neuron. And if you remember back to the beginning of my talk, we define neurons based off of the red and the green label that they have. This neuron is unlabeled. So this is simply just a cold sensing neuron. This is a cold sensing neuron which doesn't elicit pain. But up here, we have a cold sensing neuron that does elicit pain. All right, let's go back to our Constellation Pharmacology experiment, and now we're going to apply the most delicious plant-derived natural product. We're going to apply capsaicin. And this is a molecule that's produced by the spicy chili pepper. It's the compound that when you eat too much of, it becomes very painful. So now here, when we apply capsaicin, we're going to see that cell 3 responds. And up to this point, cell 3 does not possess TRPA1. It does not possess TRPM8, so it doesn't sense cold. But now I will tell you it possesses TRPV1, which means that this neuron senses heat. So let's go ahead and see what happens here when we apply this capsaicin. Cell 3 gives us a nice big response. So now the point I want to make here is that we get data from every single neuron. And here when we apply potassium, this is just going to depolarize the cell. This tells us what is a neuron. And we can see that all of these cells respond. All right, let's go back to our cell type classification. And now we understand which neurons sense heat. So the point I want to make here is that we're collecting data from thousands and thousands of cells simultaneously. Here I'm only showing you 20, but now what do we do with these thousands and thousands of traces? I've already hinted at it with our cell type classification, but we simply go ahead and sort these guys into these different groups based off of that size, that neurotransmitter, right? What type of pain does it sense? Peptidergic, non-peptidergic, or what type of sensory receptors it possesses. And so now every neuron can be assigned to one of these 16 different cell classes. Right? So we get to see what senses pain, what senses hot, what senses cold, and what senses that noxious wasabi sensation. But we still lack an identification for these types of neurons, right? At the beginning of my talk, I brought up this hyperalgesia, right? Where touch now becomes painful. How do we define that within the cell type classification? And the problem with these large, unlabeled neurons is that they look identical. They're big. They don't respond to any of those plant-derived natural products, right? So there's no response to menthol, meaning there's no cold sensation. They don't respond to capsaicin, meaning that there's no heat sensation. So how do we identify these? How do we know which of these neurons is going to sense that touch? And so luckily we turn to another source of natural products. We turn to marine-derived natural products. Here we get venom from a cone snail called Conus radiatus. This cone snail hunts fish by injecting a very complex mixture of proteins into the fish to paralyze them. If you've never seen this, go ahead and type into that YouTube search bar cone snail, or I'll simply link a video here on this video so you can actually watch this cone snail hunt. But it injects many different proteins into fish to paralyze them. One of those proteins is what our lab has identified, called kappa MR3J. And what this protein does is it blocks a voltage-gated potassium channel. I know that sounds pretty complex, but we don't have to get into it. Basically just know that this compound has a very specific target. And so we leverage this compound, this kappa MR3J, to use in our experiments. So now we can do constellation pharmacology again, right? Remember, constellation pharmacology means we leverage pharmacology to help us understand the constellation of ion channels and receptors on the surface of these cells. So by using this pharmacology, we'll understand which of these neurons have this voltage-gated potassium channel. And so that's what we do. We apply it. And what we get, this is a complex experiment, but it's very simple to understand. When we apply R3J in increasing concentrations, we get this phenotype where we get this nice smooth rise in the calcium baseline. And this is most pronounced in the second and the third application of R3J. But we also get this jagged phenotype, right? And this is very different from our smooth rise in our calcium baseline. And so now we have these two new phenotypes that we can leverage. We have this smooth phenotype and we have this jagged phenotype. And so now the question is are these cells different? They look fairly similar, right? They're big. They don't sense pain, heat, or cold. And, you know, their responses to this pharmacology are not that different. And so this brings us to the second part of the talk. How can we identify these unknown neurons with known modalities in the field? That means, how do we classify these cells and the sensation that they provide? So we turn to this technique that I like to refer to as farming. And the reason why we call this farming is because we use our pharmacology-derived phenotypes, right? The smooth and the jagged phenotype, which, let me just remind you, are classified within our cell type classification. And these cells, L1 and L2, make up less than 0.5% of the total population of neurons. This means if I have 2,000 neurons, I'm only going to see up to 10 to 20 of these neurons in my cell culture. So they're rare in our cell cultures. But using our technology, we're able to identify them very rapidly. And this is due to the cell type classification. And so now that we have these pharmacology-derived phenotypes, we can pick them up for single-cell transcriptomics, right? And what is single-cell transcriptomics? If we go back to Biology 101, we remember that DNA is transcribed into mRNA, and then mRNA is translated into protein. Transcriptomics gives us the view of mRNA. And we're going to assume for this presentation that the level of mRNA is going to correspond to the level of protein expressed. This might be a naive assumption, but so far it has worked very well for us. And so here's where we are. We've used Constellation Pharmacology to create these phenotypes that smoothen the jagged, and now we're going to use single-cell transcriptomics to tell us what these neurons are. And so this is the type of data that we get. Every single row here, L1, 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 L2, 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 these are all individual neurons. And so what I've done is I picked up 7 smooth rise neurons, and I picked up 5 jagged rise neurons. And then the type of data that we get is immense. We get thousands and thousands of genes. We get readouts of all the genes that these neurons express, as well as the genes that these neurons do not express. I'm only showing you a very small sample. I'm showing you 20 genes here, and mainly genes that are differentially expressed between the smooth rise and the jagged rise. But there are diagnostic genes that researchers have used for years to characterize a particular type of sensory neuron. One of those genes is called paravalbumin, and this gene researchers have used to study proprioceptors. Proprioceptors are the reason why you know where your arms are in space. They send these projections out to your muscles, where they wrap around these things called muscle spindles, and these little different ones are called Golgi tendon organs. But what happens is when you extend your muscle, these things are going to stretch, right? They're going to stretch and they're going to open up these channels to tell us that your muscle has stretched. And this is going to send a signal back to your central nervous system where it tells you that neuron is stretched, and likely it's going to stop firing when that muscle contracts. This neuron is the reason why you can close your eyes, and this is something you can do at home. Extend your pinky way off into space, right? And remember, your eyes are still closed. Then take your other pinky, and you can find it very easily out there in space. This is the reason why athletes can perform to the level that they can, is because they know where they are in space. And so now what we can do is we can identify these smooth rise neurons, right? We go from smooth over here, and we can now identify them as proprioceptors. So now we've leveraged our transcriptome to tell us which of these neurons, proprioceptors, which contain paravalbumin, are proprioceptors. Likewise, our jagged cells do not possess this gene whatsoever, so we're confident in saying that these cells are not proprioceptors. Additionally, we can use another diagnostic gene. This is called tyrosine kinase receptor B. And this gene, as we can see, is not expressed in our proprioceptors, but it is expressed in our jagged cells. This is a gene that researchers use to identify light touch. These neurons send projections out to your hair cells. So you guys have already seen this at the beginning of my talk. And this work was done by Dr. Jeff Woodbury and Dr. David Ginty. And what they've shown is that there's many different types of cells, but the type of cell that we've identified is this A delta LTMR. And this projects out to these hairs called zigzags. And this indicates when you feel the lightest touch on your skin, this cell is going to signal that back to your central nervous system. This is going to be really useful to tell you when there's a little ant or a spider crawling up your leg, because what that ant and spider is going to do is they're going to be touching your hairs, and this is the reason why you can feel it. And so now we've identified this jagged cell as this specific term, A delta LTMRs. But for this talk, right, A delta LTMRs is too hard to understand. So let's just refer to these guys as light touch. So we have proprioceptors, right? This is where you are in space. And we have light touch. This is telling you whether there's a spider crawling up your leg. And so now we've identified these unknown neurons with these known modalities. And so we can go back to our cell type classification, and we can now identify L1, this is a proprioceptor, and L2, this is going to tell us light touch. But this is the beautiful thing about transcriptomics, is that we have thousands and thousands of genes that we can use to predict our pharmacology. So I'm going to show you one story of how we do that. So we identify this gene, CACNA1H, and what we're going to now do is we're going to go from single cell transcriptomics here, and we're going to go back to Constellation Pharmacology. Because single cell transcriptomics is now going to give us a very good prediction for identifying cells using Constellation Pharmacology, as well as, remember, constellation means the different, helping us to understand the constellation of ion channels and receptors on the surface of these cells. And so that's what we do. We study this gene, CACNA1H. So what is CACNA1H? Well, this is a voltage-gated calcium channel. And voltage-gated calcium channels can be split into two groups. High voltage activated, these are going to be, kind of hard to activate calcium channels. And we have low voltage activated. CACNA1H makes up the low voltage activated calcium channel. Unfortunately, the field has spent a significant amount of effort in developing pharmacology directed towards these calcium channels. This small molecule, and we call it a small molecule because it's not a protein and it's small, is called TTA2. And what this TTA2 is going to do, we're going to call this an antagonist, but it's very simple. TTA2 is going to block voltage-gated potassium channel 3.2, 3.1, and 3.3. So now when we go back to my outline, we can use our transcriptome to predict our pharmacology. So let's go back to this, right? So we can see here that our proprioceptors do not have this gene, CACNA1H, but our light touch neurons do have this gene. So when we go into this experiment, we have these very specific predictions. So first off, we can identify our neurons using this marine-derived natural product, our kappa-M R3J. This is the reason why we see this jagged effect and why we see this smooth rise. And then we'll do the same thing and we'll apply R3J, but now in the presence of this CAV3 blocker. So remember, proprioceptors, which don't have that gene, they simply should not be affected. And that's what we see. We apply R3J with TTA2, and we see that this smooth rise in the calcium baseline is not affected. But when we apply it with our light touch neurons, we can see that this jagged effect is abolished in the presence of this voltage-gated calcium channel blocker. And when we go back to just R3J, we're able to recover that, and we can see that the proprioceptor is once again not affected. But the great thing about Constellation Pharmacology is we don't get data from just two neurons like we do with single-cell transcriptomics. We get data from every single neuron. And this allows us to uncover different cell types. So this figure is simply showing us that 0 out of 12 proprioceptors in that experiment are blocked by our antagonist, whereas every single L2, every single light touch neuron, 11 out of 11, are blocked by our antagonist TTA2. But here we get data from everything, right? We get data from 1,300 neurons, and we're able to see what are the other cell types that are affected. And something that was very interesting was that we saw one other cell that was affected by this blocker, CAV3.2. And out of 130 of these cells, 126 were blocked. And according to the literature, there's only two cells that have CAV3.2. There's these light touch neurons, and there's another light touch neuron called a CLTMR. And we're not going to get into it, but this is how we now leverage our results with the literature in the field to help us identify this cell type, CLTMRs. It's thought that the CLTMRs may be responsible for pleasurable touch. The gentle stroking of a mother could be activating these CLTMRs, whereas the crawling of a spider up your arm could be activating these light touch neurons. So now, once again, we go back to our cell type classification, and we can identify which neuron now senses that light mother's touch. And so N14 we've identified as CLTMRs. But now what we do up to this point is we know what happens. We know how a normal and healthy animal functions and senses its environment. But what we do is we use these cell types to help us track what happens during a disease state. And so there's been a ton of effort put into creating these disease states in animals. And the disease, well, I guess I shouldn't call it a disease state. We'll call it a chronic pain state, right? This is going to help us mimic what happened to those workers throughout America who have been afflicted by this chronic pain from an injury in the past. And so this is what we do. We do this thing called chronic constriction injury, where we tie these ligations right outside that DRG that we've been talking about. And what this does is it elicits a chronic pain state in these animals. We do it on the left here. This is called IPSI. And then we don't do anything on the right. So this means that only one side of the animal is going to be in pain. And this is going to allow us to uncover new targets for drugs. And then we use our cell types to track this disease state. Remember, this little DRG, It's going to contain 16 different cell types to help this animal sense its environment. And what we found, right, and this is our first finding, and we're finding a ton more every single day, is that this specific cell type, our R13, this is affected. This changes during this chronic pain state. And this R13 cell, what happens during this injury is that it starts to get a response to this compound called ATP. And why would a cell need to respond to ATP? Well, if we go to Cell Biology 101, ATP is what the cell uses for energy, right? So the cells are going to be chock full of this ATP. But when you damage those cells, all of a sudden that ATP is going to spill outside of the cell. And these neurons are going to be there to detect it. And what's happening is that these neurons are becoming more and more sensitive to this molecule ATP. In addition to this, we have another type of molecule called bradykinin. This is a molecule that, just like ATP, helps us to sense cell damage. And here we can see this type of neuron, our R13 neuron, is gaining a response to bradykinin. And so that's how we work, right? We're identifying and helping us to understand which neurons change during this disease state. And I think this is where it gets really interesting, because let's go back to our hyperalgesia, our light touch hyperalgesia. What happened to my father during that injury? Why all of a sudden is light touch more painful? And so what we can do now is we can begin to track what is happening to this cell type during these disease states. What about the women what about the patients who are afflicted by breast cancer who now have this cold allodynia, right? This means that a normal cold sensation is super painful. Well, we have two different cold sensing neurons, and now we can really start to hypothesize around what's going wrong. Is it this normal cold sensing neuron that now is transmitting a painful sensation to our CNS? Or is it this normal pain neuron that senses cold, but now the threshold is much lower? These are the questions that we can answer and that we are answering every single day in our lab. And so let me just go ahead and end here with our acknowledgements. This work is done by a lot of very talented people. I work for Toto Oliveira, and I've been working for him for the past 5 years. What I mainly do is I leverage computer science, engineering, and biology to help us track and uncover these problems. And the main point that I want to make here is that this approach and this strategy isn't just applied to this pain. This can be applied to any type of disease state, and it's powerful because we can track individual cell types during these disease states. What happens during Parkinson's disease? What happens during multiple sclerosis? What about Alzheimer's? What are the cell types changing? And is there a cell type that's responsible for these chronic pain? For these chronic and serious diseases? So I hope that you will join us in trying to answer these problems, and I'll be keeping you posted on the progress that we make. So thanks for listening. And once again, these are all the people that are doing it. Please look them up on LinkedIn to get more answers on what's going on. But yeah, thank you very much.